people. How are y'all doing today? Y'all? Did I say y'all? I'm not from the South. Well, kinda. I'm from Pennsylvania, which is south of here. DiGiovanni shows. Life is good. Claire, I can always count on Claire. Yay, Claire. 11:01. What do we got here? 28 people. Let's wait a couple minutes. Let's have a discussion here. Let's discuss the fact that today is 4/20 and marijuana is now legal in New York State. Thoughts?
I didn't even know marijuana was illegal or legal.
I think it's legal now. The joke has been that Cuomo made it legal so everyone would forget about all his troubles. Could work. Works for me. Anyone else as stoned as I am right now? I am not stoned. So I'm hoping you're all as stoned as I am right now. It's good for you every now and then. What the hell? Moderation, right? Right, Lake?
Definitely.
Bailey Lyons, every time I see you, your hair is darker.
Well, it's wet, so.
Oh, it's wet. Okay, okay. Have you dyed it at all, or is it just dark? It's gotten darker.
No, not since November, but my natural hair color is brown, surprisingly.
Oh, okay, okay. Kind of like my wife. I always thought she was a blonde, and after we got married, I looked at it closer. I'm like, no, that's brown. She was like a tow-headed blonde when she was born, but it got darker. It gets darker over the years. Some people's hair gets lighter over the years, which is a different process altogether. Okay, what do we got here? 29 people. Why don't we just go ahead and get started? And I'm not sure how long today's lecture is going to be. I've got two main things I want to talk about. They're both a little bit on the complicated side, so we'll see how it goes. So we will start today here. Oh, no. We're going to start talking about the exam because I finished it last night, thank God. So let's go over the exam. Yeah, so the last two essay questions I graded, I didn't put any comments down because, oh God, I just had to get through it. You know, it was taking me forever to get through it. So hopefully if you didn't do well on one of those two questions, you'll pick up today what the problem was. If not, send me an email and I'll give you better discussion on it. Bum, ba, ba, bum, bum, bum. Come on, baby. Okay, so question one. Match the type of hormone released to the physiological process. Physiology process should be physiological. An epithelial cancer cell releasing and responding to FGF. So the same cell is responding that is releasing.
Can't see the question.
Question?
We can't see what you're seeing.
Oh, Jesus. I always do this. I'm sorry. I'm kind of like a toddler. You know, a toddler says, "Daddy, look at this," and doesn't show it to you, just looks at it themselves. Like I can see inside the toddler's head. Sometimes I'm a toddler, and that's okay. Adrenal glands releasing epinephrine. That would be endocrine. Lymphocyte expressing FAS ligand. Touching should be this protein to the FAS receptor ligand, juxtacrine. Clearly, I need an editor. And that leaves paracrine, which is the Speeman organizer releasing chordin into the embryo from the dorsal lip. Okay. Hopefully that was easy. Question 2, matching. I always put this up here because it asks me to give directions for a matching question. And it's like, you match them. What do you expect? I don't know. I'm just getting cynical. The decrease of a drug's effectiveness over time. Why, that would be tachyphylaxis. Phospholipase activated by GQ. That would be phospholipase C. The transcription factor activated by the TGF-beta receptor pathways. That's Mr. SMAD or Miss SMAD. Protein that binds calcium and activates other proteins. And that would be calmodulin. An enzyme that is activated by nitric oxide in smooth muscle cells. That would be guanyl cyclase. Okay, a lot of signal transduction here. Life is good. Okay. Kinase associated with the JAK-STAT pathway. The kinase is the JAK. Intracellular adapter protein in the Wnt pathway. Oh, that would be Mr. Disheveled. Transcription factor within the JAK-STAT pathway. That is the STAT. Kinase activated by cyclic AMP, PKA. Kinase activated by diacylglycerol. That would be PKC, I believe. Yes, indeed. Receptor for Wnt. That is frizzled. Kinase activated by PDK1. That's AKT or PKB. Kinase activated by RAF. That would be MEK. And kinase activated by cyclic GMP, that's protein kinase G. Let's make sure I got them all right. And I did. Okay, good. Life is good. Number 4, embryo development. Embryo development. Hormone responsible for induction of ventral mesoderm. Ventral. BMP4. Transcription factor that produces dyspemin. Transcription factor, that would be goosecoid. Where are you, goosecoid? Embryonic tissue that will develop into skin and neurotissues. That is ectoderm. The first stage of the xenopus embryo that has 3 axes. First stage. That would be the gastrulum. Stage of the xenopus embryo only has 1 axis. That would be Mr. Blastula. Blastula. Diffusible factor that blocks the action of another hormone. Noggin. Embryonic tissue that will develop into muscle, bone, and blood. Mesoderm. Embryonic tissue that develop into gut. You haven't got the guts. Transcription factor that produces the Newcoop center. That would be CM1. Okay. Wow, I'm doing well. I think I'm going to get an A on this test. Okay. RTK. A growth factor binds to the RTKs and they transphosphorylate, which recruits blank to bind to the SH2 domain. That would have to be GRAB2. And that brings with it SOS, which is a GNRP that activates RAS. This protein activates the kinase RAF, which then activates MEK, which then activates MAP kinase. The final kinase in this cascade phosphorylates June, which binds to FOS and becomes AP1. So there's our RTK pathway. And for the last, fill in the blank, a growth factor binds to the RTKs and they transphosphorylate. Wait, I did this one. I hope I didn't put that in twice. Oh. I was being tricky here because they both start out the same way. A growth factor binds to RTKs and transphosphorylate. This makes an SH2 domain that attracts PIP3K. This enzyme phosphorylates PIP2 into PIP3. This PIP3. This phospholipid is a binding site for proteins with a pH domain, plextrin homology domain, and the enzyme blank is attracted to it. This enzyme is phosphorylated by, okay, so both of these would, here's where it gets sneaky because both of these would cover for this one, but if you've already used this one over here, then you can't use it over here. So this one has to be over here. And that means that this spot is only good for that one. And that's going to lead to beta catenin. Okay? Make sure I got that right. Yes, indeed. All right, so now essay questions. Getting used to nicotine. Okay, so there are really kind of three questions here. A person who smokes for the first time, it's funny, I wrote this question based on an experience I had when I was like 14 or 15. My mother came home from the grocery store and they're giving out free samples of Workhorse chewing tobacco. And I was from out in the country, so this is something people would do. So I said, Mom, can I try this? She was like, sure, go ahead, because she probably knew. So put a little cheek between my cheek and gum there, chewed on it, tasted like crap. Kept chewing it though, spitting, never swallowed, kept spitting, walking with my friend around town, which I mean, my hometown, walking all the way around the circumference took about 15 minutes. It's not a big town. So we're walking around town there, get about halfway and I'm starting to feel kind of woozy. Oh, what the heck is this? I'm not feeling so good. So I said, Ken, let's get a soda. So I spit my stuff out. We go to the bar across the street from my house and we get 7-Ups. We sit on the thing outside and drinking our 7-Ups. I'm like, oh, Ken, I really don't feel good. I'm going home. So I went home, laid in the bed, and the bed is spinning and spinning and spinning. I'm like, oh, and I grabbed my garbage can and emptied it. And I swear to God, in terms of volume, I've never thrown up so much in my life. Oh my God, I think gallons came out. I could feel like last week's dinner coming up. So me and nicotine don't mix, but this is really what it's about. It's like that first dose of nicotine, I got a really, really heavy dose and it was like, whoa, nicotine's a toxin, really. Okay, so first time severe effects because all your receptors are normal, your receptors that are at a normal rate, and now you're overstimulating them and you get the bad effects. So someone who smokes frequently someone who chewed tobacco frequently could have done that and would have felt nothing. Why? Because they're constantly doing it, so they're down regulating their receptors, and so it doesn't overload their receptors because they've got fewer of them. But if that person stops chewing tobacco or smoking cigarettes, then they go into withdrawal. Why is that? Well, because their receptors are low now, and if you don't have that external nicotine stimulating them, you don't have enough receptors there to get you up to normal. And so you go into this withdrawal, which means we need nicotine because our receptors are understimulated. So that's what I was looking for, those three different things. The receptors are normal, and so they're overstimulated, they get downregulated, and then once they're downregulated, you need that drug to stay normal. DS4532. This is an old question. I ask this a lot. Okay, as a pharmacologist, you're researching the effect of the new drug DS4532 on cyclic AMP levels in isolated ventricular cells. So we're just looking at cells here. DS blah, blah, blah alone increases cyclic AMP levels slightly. So we see this here. Here's what epinephrine usually does, increases cyclic A up to that high. And then you wash it out and it comes back down to here. Now you add this drug, it goes up a little bit. Not a lot, a little bit. But then you add epinephrine on top of it, and look how much higher compared to epinephrine alone. So it's potentiating the epinephrine effect, okay? It's not directly causing a whole lot of cyclic AMP production, but it's potentiating the normal pathway. That's key. And then you wash the epinephrine out, and cyclic AMP lingers for a long time. So rather than just going away quickly when you wash out epinephrine alone, with that DS4532 there, the cyclic AMP amount lingers a long time. So what could be causing this? So number one, you're going to think about your cyclic AMP pathway, receptor, G protein, adenyl cyclase, and then you've got cyclic AMP. Now, don't worry about protein kinase A. That's downstream of cyclic AMP. So we don't even care about that. That's not part of this question. But cyclic AMP then gets broken down by PDE, phosphodiesterase. So what I was looking for, which is the most obvious answer, is phosphodiesterase. If you block phosphodiesterase, then your baseline level of cyclic AMP is going to come up a little bit, but you're going to potentiate that epinephrine because now there's nothing to break cyclic AMP down. So when epinephrine brings it up, it stays up and it gets really, really high. And then when the epinephrine goes away, it's going to be broken down really slowly because you're blocking that PDE. I mean, you won't be 100% blocked, so it will come down, but you're going to potentiate it. So that was the answer I was looking for. But there could be other answers that could be really anywhere on that scheme there. So if you're talking about allosteric interactions, I think the best secondary answer, which people got credit for, was it could be bound to the G protein GS and inhibit the what do you call it? The hydrolase activity of the GS. So GS breaks down GTP a lot slower. Okay, so that means that once you activate that alpha subunit, it stays activated a lot longer than it normally would. That would increase your cyclic AMP. Or it could be some kind of allosteric interaction with adenyl cyclase. So it stays active longer when the G protein is attached to it, or makes it grab that G protein stronger or something like that. That would make more cyclic AMP. Something like that on the receptor would work as well. Maybe make the receptor have a higher affinity for the epinephrine, some allosteric site that potentiates the epinephrine binding. What wasn't going to get you a lot of points was saying that the drug directly activated the G protein or directly activated cyclic AMP. Because if it did, when you add the drug here, it would go really high, right? You get the same effect as epinephrine probably. So if you're fully activating adenyl cyclase, you would get this. And you're not, you're just getting a little bump. So the key to knowing how to answer this question is potentiating, but it's not directly activating that cyclic AMP production. That was a very pharmacological question. I think you should all become pharmacologists like me. Okay, this question, I don't know if you can see the names of the question, but I call this one, "You Making Me Spielmann." Oh, I'm silly. Okay, describe or draw a figure to explain how the Spielmann organizer creates a dorsal ventral axis. Start at the point where Spielmann organizer is already formed, not before. So I put that "not before" there on purpose because so many people will start back at sperm hitting egg, and it's like, oh, I gotta read all this. Oh, I've got 50 essays to read. Don't make me read all this. So when I say, start at this point, not before that point, you're just saving me trouble. Okay, so basically you've got a Spielmann organizer. What does it do? It releases chordin, noggin, follistatin, and XNR3, and these diffuse away from that dorsal lip towards the ventral side of the embryo. So that gives us a gradient of these three hormones going from dorsal to ventral, and these hormones are blocking the activity of BMP4. BMP4 is important for making mesoderm, but it's only ventral mesoderm that it's making, not dorsal mesoderm. So because you've got a gradient of the BMP4 blockers, you end up with a gradient of the BMP4 activity from ventral to dorsal. So where BMP4 is highest, it becomes ventral mesoderm, blood and heart and kind of things that are in the ventral side of your body. And then where it's the lowest, or really no BMP4, it's dorsal mesoderm. So that would be notochord and structures like that. Okay, so that's what I was looking for. And again, this was not anything out of the ordinary. This was just memorizing what we talked about in class. Now the next one is a little more tricky, and a lot of people didn't get the next one, but it's a hard question. You have to let me ask at least one hard question. Okay, this was your hard question. So what do you think would happen if you took an animal cap and sandwiched it between endodermal tissue and Spielman organizer? What would happen to the ectoderm from the animal cap? Okay, so now I just want to know, look, if you read the question, it says, what happens to the ectoderm? A lot of people were telling me about a second axis, and it's like you can't get an axis out of this because you've just got a chunk of ectoderm and you don't have like an embryo anymore. You've taken the whole thing apart and put it together differently. So you're not going to get an axis because you're not going to get any gastrulation out of this. It's all kind of mushed together. So all we're looking for here is what hormones are being released onto that ectoderm? What are they going to do to it? So your endoderm is releasing BMP4 and the 3 XNRs, and that's going to make mesoderm, but ventral mesoderm, right? That's only going to make ventral mesoderm because you've got the BMP4 there that makes that forces it to be ventral mesoderm. But on top of it, you've got the Spielman, which is releasing a blocker of BMP4. So all you've got, the only activity you've got now are the XNRs, and they're going to make that into dorsal mesoderm. So that's mainly what I was looking for, was that the endoderm is going to make that mesoderm, but then the Spielman organizer is going to make a dorsal mesoderm. Okay. So just looking at the hormones and what they would do in that situation. All right. Any questions? Anyone want to chat me a question here? What was, I don't know what the class average was in that test. Usually I look that up, but on Moodle, I don't get around to it usually. I think everyone did pretty well though. I don't think it was a hard test. How many people thought that was an okay test? Nobody, just me. Just me? Okay. Well, I'm the only vote that counts here, so. All right, so I'm a jerk. I know it. I know I'm a jerk. My wife tries to fix me, but you can't fix that, really. Okay, so let's do a tad of review from the last class and then move forward from there. So, so we talked about the activation of MPF, which is when your mitotic cyclin B comes together with CDC2 or CDK1. They come together, they form this complex, it gets phosphorylated multiple times and then dephosphorylated, and that's a feedback stimulation event. So you get a very rapid activation of MPF, and that makes this MPF activity rise really steeply. And when it gets to the peak, and this is important because this is what we're going to talk about today, when it gets to the peak, MPF then is going to activate a ubiquitin ligase, and that will cause the cyclin B to be ubiquitinated and then degraded, and it will go away, and that will allow the cell to come out of mitosis at the end of this. So what is MPF doing? I don't know if I said this last time, but I probably did. So MPF, what it's doing is, number one, phosphorylating those nuclear lamins, which are intermediary proteins that are holding the nuclear envelope together. They're phosphorylating condensin, which is causing the condensation of the chromosomes. So they're going into that X-shaped structure, causing mitotic spindle formation. So all these things are what prophase is, right? This is what prophase is, activating out these 3 different processes. So it's directly responsible for the cell going into prophase. And then when MPF gets degraded and turns off, all these things reverse, which is telophase, right? Nuclear envelope goes back together, the chromosomes decondense, and the mitotic spindle breaks down. So MPF is directly responsible here for mitosis. And then the third thing it does is targeted protein degradation. So MPF is going to activate another protein complex called APC, anaphase promoting complex. And this degrades that cyclin and allows mitosis to end. And another thing it does is it's going to be controlling that transition from metaphase into anaphase. So if you remember, I know you remember, but I'm going to tell you anyway, because I need to kill time. Okay, right here. Remember, we had this checkpoint, which are very similar. They're cyclin and CDK dependent. This checkpoint is very different. This is your metaphase anaphase transition. So I'm going to start today talking about how that happens. Okay, so we've got a number of proteins here that are important in that metaphase anaphase transition. Number one is anaphase promoting complex. So it's a complex, which means it's multiple proteins put together. And it is the ubiquitin ligase. So this is what, this is what is going to phosphorylate ubiquitinate cyclin B and a lot of other proteins. As we'll see, it'll be important for breaking this connection between the sister chromatids. So kind of the point here of this, I should have said this before, the point of this checkpoint is that none of the chromosomes will separate until they're all connected to the mitotic spindle. And this is what makes sure that they remain, that mitosis doesn't go to anaphase until all the chromatids are connected to the mitotic spindle. So ubiquitin ligase, we'll see this will be important in that separation. Keep in mind here, we talked about in the GSK3 beta pathway, we had an APC as well. That's a different protein. That's Adenomatosis polyposis coli. So that's a totally different protein. Don't confuse that in your mind with this APC. This is anaphase promoting complex. Okay, so it's a complex. What activates it? CDC20 activates it. Okay, so CDC20 is going to bind to APC and make it fully active. Now, another thing that's important for APC activity is phosphorylation by MPF. So I said earlier, MPF causes its own degradation. This is how it does that. So MPF phosphorylates anaphase promoting complex, which kind of primes it. And then when CDC20 binds to it, it's fully active, and then we get through that transition. How does it get fully, how does this CDC20 get there though? Normally CDC20 is bound to the kinetochore of the sister chromatids. So it's not going to be released until they attach into the mitotic spindle. So MAD and BUB proteins are keeping that CDC20 on that onto that kinetochore until the mitotic spindle attaches to it, and then the MAD and BUB release CDC20, which goes off and activates APC. Then cohesins. Cohesins are proteins that bind the sister chromatids together. So right at the center of all your chromosomes, there's like some DNA that is like, I think it's like inverted repeats or something like that. So the DNA itself kind of meshes and holds those chromatids together. But more than that, what's holding them together is these cohesin proteins that are attached all around that kinetochore. And that's making sure that as the mitotic spindle pulls it in both ways, and there's a lot of tension on these things, they're not letting go because those cohesins are holding them together. And then we've got this enzyme, separase. Separase is a protease. It's going to break down the cohesins, and that causes you to go into that metaphase, from metaphase into anaphase. So separase kind of ends that checkpoint. And then finally, securase is going to block separase. So separase isn't active until we activate APC. APC is going to ubiquitinate securase, break it down. Then separase is active, it breaks down the cohesins, and then we separate. So just to go over this real quick, and then we'll do it again in a second. When you get to that metaphase transition and those sister chromatids are stuck, they're going to stay stuck like that until the microtubules attach to the kinetochore, and that all the kinetochores, and that allows the MAD and BUB proteins to let go of CDC20. CDC20 then binds to APC and activates it. It's already been pre-activated by MPC. It then comes down, ubiquitinates securase. Securase gets broken down. Separase breaks the cohesins, et voila. And then they open. So let's see a figure on that. Here we are, MAD and BUB proteins in this kinetochore holding this all together here. Where's our CDC20? CDC20. So these are inhibiting CDC20 at that kinetochore. And then when they are connected, they release CDC20. Okay, so when the kinetochore attaches to the mitotic spindle, they release CDC20. It comes down and activates APC. APC then degrades securin and activates, and which releases separase. Separase breaks down the cohesins, and anaphase proceeds. And at the same time, this is breaking down that mitotic cyclin. Okay. Oh, what the hell? Okay. So there's a really cool animation on this by my personal animation god, Drew Barry. So you know we're going to watch this animation. Let's watch this. Okay, volume up. Open.
Your cells is just 2 nanometers across, but around 30 to 40 million nanometers long. To keep DNA organized and regulate access to the genetic code, it is wrapped around structural proteins called nucleosomes, which are then further organized into fibers called chromatin. The chromatin fibers are packaged into gigantic molecular structures called chromosomes. We are now pulling away from the chromosome, out through the nuclear core, which is a gateway through the nuclear membrane. We are zooming back to reveal a cell about to undergo mitosis. The nuclear membrane breaks down, releasing the chromosomes. The chromosomes are arranged in the middle of the cell before being pulled apart. Identical sets of DNA going to each side. The cell then splits down the middle, creating 2 new daughter cells.
Let me just pause for a second. So something he doesn't talk about here that I think is kind of neat to see is I said when the cell goes into prophase, the cytoskeleton breaks down and the cell rounds up. Now you can see in telophase how after mitosis, the cell flattens out back to its normal interphase shape.
See the chromosomes. The chromosomes are arranged in the middle of the cell before being pulled apart with identical sets of DNA going to each side.
So now watch the 2 cells flatten out.
The cell then splits down the middle, creating 2 new daughter cells.
And that flattening is the interphase cytoskeleton setting up again.
This visualization isolates a single chromosome at mitosis. The chromosome contains 2 identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatins. Sticking out on either side are microtubule fibers from the mitotic spindle used for orienting and guiding the chromosomes to their correct positions. The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system which is able to feel everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension is just right, and the chromosome is correctly positioned and attached. The last little bit remaining role is the stop signal broadcasting system that is carried away by a dynein motor walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding to the kinetochore on long molecular legs are dyneins carrying the stop signal broadcasting system.
That's pretty awesome.
When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
So he doesn't get into the Mad and Bub stuff. The next animation I want to play for you does. But I kind of want to watch this one. This one's really cool too. I hope you don't mind. This is replication. Oh, gene activation. Look at this. Oh, no, I'm sorry. It's histones. Wow, look how that thing winds up, how tight it is. Three different levels of winding there. Guess I was wrong about the replication part. Well, oh, here's replication. Remember your leading and lagging strand of replication. This one's doing the lagging strand over here. Boom, hits the end, stops, comes around, starts again. Here's your leading strand. It just keeps going. Just keeps going. So this is doing the lagging strand over here. This is your leading strand here. This Drew Berry is a god. This is so beautiful. I love the sounds they add too. Like it's a big metal structure clanging around. My God. Isn't that the most beautiful thing you ever saw? All right, let's stop this one here. Get back to what we were supposed to be talking about, Mad and Bub. So let's watch this. How long is this? 2 minutes. Okay, that was a tad boring. That was definitely not as good as Drew Barry. Did I call him a god yet? I think I did. Okay. So, a second. Rearranging my landscape again. Take a quick look at my notes to make sure I didn't forget anything. Oh, Dr. Slish, you never forget anything. Oh, come on now. You know I do sometimes. I swear to God, I'm becoming a nutcase here in this room all by myself. I just keep talking to myself. And then every now and then I'm like, wait a minute. You know, there are people listening to you. Oh, shit. You're right. I should mute my audio. Okay. All right. So that's the metaphase anaphase transition. Any questions or comments about that? I think it makes sense. Okay, so just to kind of sum up, we've got these three different checkpoints. This one is caused by this cyclin B and CDC2 and the phosphorylation, dephosphorylation, et cetera. This is the, excuse me, this is the one we're going to talk about next, which is a different cyclin and a different CDK, the G1 cyclins, which are cyclins D and E, and G1 cyclins, which are 4, 6, and 2. Okay, so these are going to be similar. There's also phosphorylation, dephosphorylation events that happen with this. So, and then this checkpoint's different because it's that anaphase promoting complex checkpoint. Okay, so what we're going to talk about next is how it is that we get these G cyclins, because remember, this is the restriction point that tells the cell to go forward. And this is the only point in the cell's life where it's responding to external signals. So the external signal is a growth factor, which tells the cell to go ahead and go through that restriction point. And after that, it's totally insensitive to growth factors until it gets back into G1. So what we want to talk about now is how those growth factors actually get the cell to go through that restriction point. And it has to do with a couple of really interesting mechanisms. So here's this. Just reviewing, I just did this in the class today. You've got RTKs that activate GRAB2 and SOS. SOS activates RAS, RAF, MEK, MAP kinase, AP1. So what does AP1 do? I'm glad you asked, because this is what's going to get us into the next phase. Hang on one sec. Pull up my other set of notes to make sure I'm not forgetting something. By the way, I need to re-upload my notes because the slide numbers are wrong at this point. Okay, so I don't think I have a slide on this, but I need to talk about it. All right, let's talk about it by looking at my stop share. New share. These notes are uploaded there, but they're labeled as lecture 3. So I'm going to change that after class. Okay, so we pick up the story. When we left our hero, he was phosphorylating June to create FOS and made AP1. AP1 then is going to turn on, is a transcription factor, and is going to turn on a whole series of genes. And by the way, AP1 is not the only transcription factor that gets turned on here. There's another one called ETS, and there are several other ones that get turned on. So we always kind of like bring it down to something simple so it's easy to understand. And once we get into biology of cancer, we may expand that to include more detail. But right now, this is probably enough. So anyway, the whole transduction pathway activates AP1. And this is going to then turn on a whole range of genes, and we're going to call these immediate early genes, IEGs. And they're called immediate early genes because they come on within 30 minutes of stimulating the cell with the hormone. So they discovered this by taking cells in culture, and they just look at what genes are on when it's in that G1 state. And then they hit it with the hormone, and they follow gene transcription, and they see that this whole bunch of genes comes up is 30 minutes after you hit it with that hormone. So these are immediate early. And here's another interesting point. You could hit these cells with a translational inhibitor like cyclohexamide. Cyclohexamide is the classic drug used to block translation. So you want to know, the question here is, do we need to have translation to have these genes turned on? Or is all the machinery necessary to turn them on there already? And what we find out is if you take these quiescent cells, treat them with cyclohexamide so there's no transcription, and then you hit them with the hormone, all these immediate early genes come up, which tells you everything needed to turn them on is there already. We don't need any protein synthesis to make that happen. So these immediate early genes then are the ones that are just going to be the first ones that are turned on. So what do we get? Some of them are transcription factors. Not surprising. Everything's like a cascade process here. So Jun and Phos make AP1, and that makes more Jun and Phos. So we get a lot more of this thing coming up. And we also get this guy, Mick. Mick is pretty amazing in that it alone can make the cell go into cell division. It does so many important things. We'll learn about this in biology of cancer if you're in that class. But Mick by itself can cause the cell to divide. So these then get transcribed. They go out of the nucleus, they get translated, and then they come back into the nucleus and they turn on transcription of another wave of genes. So these are going to be called delayed early genes because they're delayed by 60 to 90 minutes because those first set of genes have to turn on, get transported out, their RNA is transported out, translated, proteins get transported back in, and then turn on transcription from those genes. So what you see in this experiment is mediate early genes come up, then they go down, and then delayed genes come up. And the delayed genes are what's actually going to cause the cell to start to divide. So if we go back to that cyclohexamide experiment, your immediate early genes come up, and then you never get delayed early genes because you need that translation to make those delayed early genes come on. Okay, so that cyclohexamide experiment is really important because it shows you what is necessary to make this happen. Okay, so what are our delayed early genes then? E2F, cyclins D and E, and CDKs 4, 6, and 2. So I said earlier, and I wasn't lying, that transition through the R is going to be caused by these CDKs that are interacting with these cyclins. So cyclin D interacts with 4 and 6, turning them on, and that gets you from G1 or G0 up to the restriction point. And then CDK2 and cyclin E gets you through that restriction point. So E2F then is another transcription factor, and it is going to be activated by cyclin CDK2 and cyclin E. And then once it is activated, it will cause the cell to go into S phase, and that is the actual event that puts you into the cell cycle into S phase. But you see, it's transcribed at the same time as these, but it's not on when it's transcribed because as soon as it's made, E2F gets grabbed by another protein, RB. RB is called a tumor suppressor because it's going to stop the cell from dividing, okay? So it's going to hold that E2F and block the passage until your cyclin and CDK complexes are active. Once they're active, they will phosphorylate RB and inhibit it, and then that will cause the release of E2F, and then the cell goes into S phase. Okay, so this is the complicated stuff I want to talk about now. Let's see what my figures are like at this point. What is this? Chapter 8? Oh boy. That is the wrong class. Let's try a different one. Okay. There we are. Okay, so this is that RBE2F thing, okay? RB is a protein that binds to E2F. So delayed early genes get activated, you make E2F, RB grabs it, so it can't do anything. And then once you get CDK and cyclin coming up, they will attach to each other and they phosphorylate RB. RB lets go of E2F, and then you go into translation. I'm sorry, you go into S phase, replication. So these proteins that are going to get turned on are things like DNA polymerase, helicase, telomerase, topoisomerase, single-strand binding proteins, all the proteins that are necessary to replicate your genome. That's what's being turned on here by E2F. Turn this stupid thing off. I hate to turn off my phone because then I lose it and I can't call myself to find it again. Okay, so let me talk about this again, and then I'm going to let you go. This is about as far as I want to go today. So I used to have a nice animation for it, but again, it doesn't work now. But I think Claire told me how to make it work. I just haven't gotten around to that yet. Okay, so you got a cell in G0, right? It's, it's doing its job, whatever. It's like let's say it's a fibroblast cell that's crawling around looking for any damage. Boom, it finds damage, it gets activated. And the activation is the fibroblast releasing fibroblast growth factor and then responding to it. And it responds to it by activating this pathway. So FGF binds to the RTK, which activates all the way down to MAP kinase. MAP kinase makes AP1. AP1 will then activate those immediate early genes. Some of them are MYC, FOS, and JUN. They go back out, they get transcribed or translated, they come back in, and they activate production of E2F and these cyclins and CDKs. E2F is going to be bound, and that's what the restriction point is, RB bound to that E2F, stopping it from working. The cyclin and CDK then will do what they always do. Cyclin comes up, binds to CDK, gets phosphorylated, and then gets dephosphorylated. Now it's active, and it phosphorylates that RB. RB changes conformation. Let's go to of E2F. It binds to the S phase enzymes, and then off it goes into cell division. Okay. Comments? Any discussion related to this that we need to do? Carrie's the person who told me how to make my animations work again. Maybe I'll work on that this afternoon. Okay, any comments? Nothing? You got nothing for me? No one wants to ask any questions like about where's our daily quiz? No one said anything about a daily quiz, which we always have. No, nothing? Well, we've got a daily quiz. This is something I've been doing, like surprising people with the quiz, because I noticed in my last developmental class that I open the class and I've got 32 students in it, and I go, oh, sorry, no quiz today. 5 people leave. Oh, really? So I put a quiz together really quick to benefit the people who showed up. So I do have a quiz for you today. I need to find it. I've got to find it. I've got to find my quiz. You wouldn't know there was a quiz, though, because I hid it. That's right, I hid it. And I'm going to unhide it now. And once I unhide it, you will have 3 minutes to do it. So everybody go to Moodle right now if you want this bonus quiz. Aha. You see? Right here it says daily quiz 420. It is hidden. If everybody's ready, I'm going to unhide it, and you can go ahead and do that quiz. Swear to God. Boom. Okay, it's up. You got 3 minutes. Rock-a-rolla. And then I'm just going to go away. So actually, I'm going to go away now. So go ahead and do that quiz, and we will talk again on the morrow.